So the title for this presentation is Controlled Release. But then one can ask, is this important for WWSC and research? Let's take a look. The first question I want to pinpoint is, what is the similarities between trees and tablets? Obviously, both starts at T, but that may not be so much to come with. Instead, I can say that both systems have very critical water transport. Here's from the road system, and here is sap that comes out from the birch. And in the tablet system, we always need to have water to come in, otherwise we will not get any release. Both system contains cellulose. Cellulose is the most common excipients when it comes to oral controlled drug release, or oral release, I should say. And both contains pore structures. And we will now talk about these different aspects, but I will take them from a pharmaceutical point of view. Is it something strange, Linda, or can I continue? I continue.
You can continue. Thank you.
So I want to a little bit start with changing the first title to Oral Controlled Drug Release. So I start with oral controlled drug release, and then I want to connect it to the WWSC. But at the same time, I want to get a warning here, because I will use the terms that we have and we use in the pharmaceutical technology industry. So it might turn out that I will use words that you recognize, but you don't actually use them in the same way as I do. But I have decided that I should stick to the way pharmaceutical technology use them. So, and hopefully we can discuss it later, and we can also, you can understand what I mean anyway. So the first question is, why do we at all have oral administration of drugs? Yeah, here's the body. We have two different types of ways that we really want, the principles we want to give the drugs to people. And one is that we would like to have a local administration. So for example, if you have a skin and then you would treat the skin. So that is a local administration. The other one is a systemic administration, where you, for example, you can't give a local administration to the heart. Therefore, you need to take the drug orally, and it starts to dissolve in intestinal, and then it is transported from the dissolved drugs is transported in the lumen here. So this is the small intestinal, where we have the villi and we have the microvilli, and this creates a huge area, super good, which then enables the drug transport very efficiently to the bloodstream, and the blood then takes the drug to the heart. So that is where we have a systemic approach of drug delivery. So the concentration then in the blood will then determine what effect we will have. And here, if you have too high concentration, then you will get side effects, bad. And if you don't have too low concentration, you will not get an effect at all, also bad. So we have a range here where we really want to have the blood concentration of the drug. Then comes a problem. Many drugs that you take will then have a very short half-time in the body. So you take them, the concentration increases rapidly and then decreases due to that you have a very efficient elimination, for example, in the liver here. So we have a system here where you get side effects and no effects, but still the patient really needs the drug. So then they take another one, get the side effects and no effect again. And then it ends up during 24 hours, you might have taken 4 different tablets. This is not so good, because that patient compliance is very low if you need to take this amount of tablets. Therefore, one have developed something that is called extended controlled release, where you still have the same drug with a short elimination, very fast or very short lifetime, but you then control the blood concentration by the release from the formulation, and then you can keep it within this range over 24 hours, which means that after 24 hours, the patient need to take another tablet. But then they have a habit, they take it to the breakfast or something like that. That will then enable much better patient compliance, which is the important here. Otherwise, you don't get an effect of the drugs. Drugs in the bottle on the table give no effect to the patients. So therefore, we have really wants to control the drug release. So there are different principles that one can do this with. So here, and these are the three main principles. So you can have a system where you have a membrane, and this membrane is black and insoluble. So here I have illustrated insoluble polymers with black. Inside you have the active pharmaceutical ingredient, or what I call API. And this can be a tablet or pellets. So this is one of the examples where I will talk about. I will not talk about this insoluble matrix tablets, but it is you have the black is insoluble, and you have the drug, and the drug dissolves and goes through these channels out and is released, and that will then be, so the skeleton here of the insoluble matrix will be very good so one can get this release.
Hello.
And sorry. I continue. So here, this system I will instead talk about, and here I've illustrated the polymer with a blue polymer, and that means that it is water soluble. And here we have the drug particles embedded inside this polymatrix. So we start over here, and then I will use the name coated tablets. That is the way one produces them. In some literature, you will see that it is membrane systems also, but I will stick to the coated word here. And we have I will illustrate this with two different principles. So I start with this one, where you have the active substance here and the insoluble matrix, and you place it in water. And the first thing that happens then is that the water starts to diffuse through the membrane. And this then makes the APIs to start to dissolve. And then you create an osmotic pressure inside here. This osmotic pressure will expand the unit, the membrane. And then it expands more and more, and then it snaps and cracks. And you get a crack here, and through this crack, you will get a drug release. So in this case, the release rate is controlled by the osmotic pressure difference inside and outside the formulation. So therefore, we call this an osmotic controlled release. Now I will go over to the other one that I have been working more with, and that is this system where you instead have pore-forming agents here in the membrane. So when you take this formulation and place it in water, the water starts to diffuse through the membrane, but also to dissolve the pore-forming agents here. And that then start to be the pore-forming agent starts to dissolve and is released. And at the same time, you get water in here, and that starts to dissolve the API, and you will get pores here, or water-filled channels, where we before we had the pore-forming agent here. So then through these pores, the drug will be released. And here, it is mainly the difference in concentration inside and outside the drug that is controlling the release rate. So I would like to focus a little bit on these diffusion controlled systems, and I would like to that we zoom in here on one pore where we have the API in a solid phase and we have the API in a liquid phase, where it's dissolved, and here is the membrane. And we have this pore, and here is the surrounding bulk liquid. And the thickness of the membrane is H. And then we apply the Fick's law to this one. So we take the concentration inside here, the membrane, minus the concentration outside in the bulk. And that will, and divided by the H, that will give us the concentration gradient. Before I continue now, I also should remind you, when you have this equilibrium between a solid API and in equilibrium with a saturated solution here, this means that you have locked the concentration in here. That means that it is the solubility, corresponding to the solubility. So this will not change as long as you have active pharmaceutical gradient in a solid state left. Since the bulk concentration is very close to zero, we can put a 0 here, and we have then the efficient diffusion coefficient here, and I will come back to that later, and also the area of the unit. And all these factors controls that mass transport over this membrane. So if we now take a look into this one, and we can integrate, and here we plot the fraction of released drug. So this is at the time 10 t, and then here we have it at infinity. So when everything is released as a function of time, this is what we call a release curve. So what happens first is that we have a lag time here. That means that it takes time for the water to penetrate in and dissolve the pore, and also dissolve the drug, and before the drug is released. So that is what we call a lag time. Here we have a straight line, and that is depending on, if you take a look here, none of these parameters is time dependent. As long as they have a constant, we have active, undissolved drug inside here, then we will have this equilibrium, we will have a constant drug concentration here. So therefore we get a straight line, but as soon as we don't have any API solid state here left, then the concentration gradient will be lower, and the slope, that is the rate here, will then be lower and lower until everything has released. So if we then continue and look into the coating materials, they are very often cellulosa derivatives. And as you know, cellulose has 3 OH groups, and these OH groups can be substituted. And this coating can be applied to core, where the core is a tablet or the core is an pellets. But so the cellulosa derivatives then can then have many different functions. They are used for a lot of things. They can, for example, enhance the viscosity. They can stabilize colloidal systems, but this makes them very useful for many different applications, for like cosmetics, paints, and food, and so on. And as we will talk about today, coatings. So I will now illustrate this on this system here, where we have the pore forming agent, then that leaks out. In this case, I will illustrate it with HPC. So we have 2 different cellulosa ethers here. The first one is ethyl groups. This means that we have ethyl groups placed on the substituents here. And this pharmaceutical approved polymer, ethyl cellulose, has a degree of substitution that is equal to 2.5 around. And that means that on average, 2.5 of these groups per glucose unit is substituted. This gives it a very low water solubility and a very low permeability of the drug. We have the pore forming agent, that is HPC. So this is an hydroxypropyl group, and sometimes we can even get a chain of hydroxypropyl groups. This polymer is water soluble, and that is exactly the combination of polymers that AstraZeneca has used in the 80s in silicon sock. to control the release of metoprolol, which is good for hypertension and heart medicine. When you apply the coatings, one can do it in different ways. If you have pellets, it's very common that you use a fluidized bed. And I don't know how much you know about fluidized beds, but in fluidized beds, you have a pressure here of air and you have the particles, and they start to dance in this system like this, going up and down, around like this. And they pass then the Worcester column. And here we have a spray nozzle. And so here is the core with the API, and then we have a spray nozzle that sprays small droplets to the surface, and these droplets float out and wet the surface and dries. So if we take it a little bit slowly here, so here, oh, that was not so slow, but here it is when sprayed, so they got the particles, the pellets get a shower, and then they are wet here, and here they dry. Since the air stream here is high, you can control the temperature of it. So you have a drying of the droplets that is now have been on the wetted surface, and they must be dry. The pellets must be dry when it comes here, because then it will come in contact with other pellets. And if they are not dry, they will glue together and you will get twin pellets, and that is not good at all. Then next time it goes passes again the Worcester column, and it gets a new shower, and then you get many showers on top of each other, and each time they dry in between. And that is a little bit of a discussion. We will talk a little bit more about that, but theoretically they dry, and then you will get increase the thickness here of the coating. So HPC and EC are polymers, and polymers, if you dissolve them in a solvent, they are very prone to phase separate, and that depends on, so you can have them in one phase in the beginning, but with time, if you change the temperature or the composition, then you will get the phase separation. So you will get one A-rich phase, here illustrated by the dark, and one white B phase. And the B phase here is rich in B, but it contains still a little bit of A. And these molecules of A is then transported to this A-rich phase, which is not, when we talk about diffusion, normally we say that you have a high concentration and then it goes to the low concentration regions. That was what we saw before. But here we have a situation where the thermodynamics rules everything. So it is not, so we have actually uphill diffusion where it goes from low concentration to high concentration of the A phase, which is a little bit funny. And so we have a phase diagram here. So we have the ethanol up here. We have the polymer A, the ethyl cellulose, and we have the B, the HPC. And we start with a one-phase area, and we have a two-phase area here. So if we start in this one-phase area up here, and we take away the ethanol, we will end up in this two-phase region, and we will get a phase separation with an A-rich phase and a B-rich phase. And then they are kept together here with a tie line. So if we apply now this for this system where we have a spray solution that contains the two polymers and the ethanol, we spray it, and with solvent evaporates. So we start in one phase region, enters into the two phase region, and we get the phase separation. And this phase separation there can be different types of phase separation. Here is an illustration from Wikipedia on spinol decomposition. A little bit funny here is that Hilrad or something, he was born in Gothenburg, developed this theory together with Kahn at MIT, and then he became an professor emeritus at KTH. So this connects Gothenburg and KTH. But you can see here that the faces, they start very small, and then they develop, and the face regions became larger and larger. And then one can lock them in a one case. And then you can use this and lock them at different times. And that depends on, for example, the viscosity. If you have high viscosity, then you can have an arrested morphology of the coating. So here we have different times, locks them at different time, the spinel decomposition can develop different long time, and therefore it is locked at different structures. So we have the HPC rich phase here and the EC rich phase here. And now, if we take that and have that on here on the membrane, we have the API here. So we have this HPC phase here, and then we expose it to water. Then the water will dissolve the HPC, and then the drug can be released from inside the coating to the bulk solution. And here I want you to observe that we have also spots that it's not connected, the HPC regions. And these will not dissolve, and therefore will not create pores where the API can be transported through. So what we did was that we made free films. We placed them in water, and we had these structures, and with time, in water, the HPC was dissolved. And then we freeze-dried them and looked into them to characterize the structures. And here we have an increase in molecular weight. You can see here are the pores much more tiny compared to here. So this is where the HPC was before, but now it is an empty hole there instead. And the reason why this happens is that when we have increased the viscosity, we lock them more fast. So we arrest the structure earlier here during the spinodal decomposition, which is then controlling the structure of the films. So one way to study the effects of this is to use a side-by-side diffusion cell, where you have a donor and an acceptor, and in between you place the free films. And we then add a radioactive labeled water here and look how fast the radioactive labeled water is transported to this side. That we use a radioactive labeled water is a tritium labeled water is very good, because then we create the same osmotic pressure on both sides. So it is actually the fusion process we are looking into. And here we have the water, and we did the same for mannitol, and you can see the permeability for one particular film here is higher for water than mannitol. But also that when we increase the molecular weight of the EC phase, then we will get a lower and lower permeability through the film. And if we then instead take a core system where we have the API here, and then we can see that the drug release, when we increase the molecular weight, we get the slower and slower drug release rates. So the same phenomena happens if we have water molecules that should penetrate through the coating, or if it is the drug. And I should say also that in this case, we varied the molecular weight on EC. We did a similar study where we varied the effect on the HPC also. Another way that is a little bit less discussed is that one can also create different pore structures by having the processing and the creation of the coating. So in this case here, we sprayed very slowly, which means that they were dry faster, and that they had dried faster meant that they were arrested earlier in this development of the phase separation. So that gave directly an influence of the pore structure, but also on the permeability. So decrease in the spinola in the spray rate locked them earlier. And here is a slide where I try to summarize the effects of that we have on control release. So this is the equation over the membrane as we talked about. But here I have written out the parameters that the efficient diffusion coefficients is depending on. And that is inspired by Ronald Siegel, a very nice guy, who was also in the advisory board for Sumo for many years. So the efficient diffusion coefficient depends on the porosity, so how many pores you have, but also how these pores are connected. So the connectivity here, or constriction factor, and also torosity. If you have a straight pore, you have this length you should pass. If you, as you always have, bands in the pores, then they will take different ways, and that is called that they have different torosities. And the first three factor here is then depending directly on the fin, whereas the last one here is the interactions between the active substance and the wall in the pore, and of course the diffusion coefficient of the drug as well. So if I try to summarize this part, so the mass transport is depending on the phase separation, the structure we got, and that depends on the drying speed and also the molecular weights of the polymers, since they arrest the pore structure in different ways. It also depends on how fast the water can be transported through the coating and the transport of the drug out from the coating. And also, which is not that is very important in the pharmaceutical industry, that you have good enough transport over the intestinal membrane. So when you have released the drug in the lumen, in the intestinal, then you need to have it efficient transported over to the bloodstream. Otherwise, it will not give any effect. So that was what I wanted to say about this system. And now I would like to continue with the erosion control matrices. So they have then the hydrophilic polymer here, and they have the API. And an advantage with this type of systems is that you can have very different amounts of drugs. That means that you can have different dosages of the drug. And one of the most common hydrophilic polymers that is used in the pharmaceutical industry is HPC, hydroxypropyl methylcellulose. So this is the creating the controlling release parts. But to manufacture them, you sometimes need to add lubricants in the tablet machine, but you can also need to have flow additives that helps the powder to flow to the tablet machine. But we will focus on the two first here, and mainly first on the polymer. So here we have the hydrophilic polymer, and we place it in water. Then the water starts to penetrate and start to dissolve the polymer and create a highly concentrated polymer solution, or what they in pharmaceutical industry call a gel layer. And the dark blue here is what we call a core. So we have a gel layer and a core here. And if we look a little bit more onto focus here a little bit, how does the polymer chain look like? So out in the bulk, we have the polymers swims around as alone polymers. So we have a dilute polymer solution. At the boundary here, between the bulk and the gel layer, we have approximately the overlap concentration. And that is when the polymer chains starts to feel each other and interact a little bit with each other. And here, when they increase in the concentration even more, then they start to entangle in each other. And there you have the entangled polymer solution. And the overlap concentration here is a very important parameter, and that depends on the polymer chains. So a larger molecular weight of the polymer will give a lower overlap concentration. So if we go back to this picture here, we still have not discussed the drug. We focus just now only on the polymers. So you can see that the water still penetrates and continues to dissolve the core. So the core diminishes. And here on the surface, you have swelling and the water is transported into the gel layer and diluted more and more until it reaches the overlap concentration where it can is released and disentangle and erode from the surface. That we call erosion when it is released from the surface of the gel layer. And water continue to penetrate and we have no core left. And in the beginning, the polymer gel layer swells and at a certain time, the erosion time rate is faster than the swelling rate, and therefore we get a diminish of the core layer, and in the end we have no tablet or matrix left. So if we take a look here on this case, which is a little bit interesting, where we have the core here and we have the gel layer here. So here is the core, and here we have a polymer concentration that goes out here, and here we then have the overlap concentration. We should pay a little bit attention to this boundary here. The hydrophilic matrix tablets have a glossy polymer, most of them at least, start with a glossy polymer with a high TG. When the water penetrates here, then the water plasticides the polymer and the TG decreases until it reaches 37 degrees, because that is the temperature we have. And then we have a transition of the polymer going from a glossy state to a rubber state. And in the rubber state, it is much more flexible and it can expand and much more. So the relaxation time here is when it goes from glossy to the rubber state. That is the rate limiting step. This means that this core diminutions is not controlled by the classical diffusion controlled water transport. It is instead non-thickened transport. So, and that is what we call polymer relaxation is the rate limiting step. And here's an example from Farad where he made a tablet, put it in water and placed it on the table. So here we see the core, and here we have the gel layer here around it. Now we should take the drug finally. That is good. Now that is what most people are more interested in is of course the drug release. So here we have the drug particles embedded in the core, and we put them in water. We get this gel layer formation, but the API particles here is protected in the dry core because the API particles can't dissolve if there are no water there. But finally, the core diminish and we get API particles out in the gel layer, and then two things can happen. They may dissolve or they may not dissolve. So they may stay as the particles. And the funny thing is that both the dissolved API molecules and the API particles can be transported through the gel layer, and that I will tell you a little bit more about very soon. When it comes out, then the API particles or the molecules are then released. So that is how that we have a transport out here. So now we should look into this a little bit more. So here we focus again. So we have the core and the gel, and here we have start with the API particle inside the core, and we have the overlap concentration here at the surface. So the API is then protected inside the core, but after a while the core has diminished and the API particles are now out in the gel. And now we assume that we have a very soluble drug. Then it's dissolves, start to dissolve here, and you get it fully dissolved, and then the drug molecules then can diffuse through the gel layer and be released to the bulk. But if you have a particle, an API with a much lower solubility, then it will not dissolve inside the gel layer here, or some molecules will of course be dissolved, but the solubility is lower, so they are not going away directly. And here I would like that you focus on the polymer concentration gradient. On this side here, we have a higher polymer concentration compared to this side. And therefore, we have a higher swelling capacity, that the undissolved particles is then pushed through the gel layer like this, due to this concentration gradient, and then they are released outside here. And when they are released, then they will dissolve. See, like this. So this pushing of particles, that is called for translocation. And so that is a little bit unusual release mechanism. Normally it is molecules that is transported, but in this case it is actually particles that are transported through the gel layer. And now we can see that this thickness of the gel layer is very important, and that is controlled by the overlap concentration. And a thicker gel layer will then give a slower drug release rate. So the overlap concentration is a very important parameter. So you can tune the drug release rate by changing the molecular weight of the polymer chains. So we were the first one actually that were able to show that the release rate for polymers was depending on the overlap concentration, proportional to the overlap concentration. So we determined the intrinsic viscosity, and 1 over the intrinsic viscosity is proportional to the overlap concentration. And here we can see that we got a quite nice line here. So this is an indirect proof that the boundary here on the surface is directly related to the overlap concentration. So therefore, one can say that the degree of swelling and the overlap concentration is very important. So one need to determine the degree of swelling. So then one can use something called a rotating disc. So here is the metal that you glue a tablet on and place it in a release bath. Have it there for some time and rotate it. And then you unscrew the rotating disc, place it in a beaker, and you have a probe here. And this probe then can determine the force and the position. And by doing that, one can then determine where, when it hits the gel layer, the force increases. And when it hits the core, it increases even more. So therefore, you can both determine the where you have the gel layer starts and where the core starts. So that is a very good method. Another method is to take the rotating disc, place a tablet on it, and place the whole thing in an MRI. So here's a magnetic MRI function. So here we don't have any gel layer, but here you can start to develop the gel layer, and it goes on, and you create more and more gel layer, and the core is lower and lower. And this technique then opens up for that you can determine the polymer, not only where you have the core and where you have the gel layer starts, you can also determine the concentration profiles here. So we did that, and we also developed simulation models to compare and also to check what is it actually that controls the release and drug release rates. Now, so here is just an example where we have determined the position of the gel and also compare it to the drug release rate. And in this case, we use this simpler method, but still working very well, where we determine the height of the tablet. Now I would like to talk a little bit about the HPMC, which is a polymer. So of course the chain length is important, but also the degree of substitution. So how many HP groups you have and how many methyl groups you have will control the behavior in the water and also the overlap concentration. But here's another factor, that is the substitution pattern. And we have looked into the substitution pattern along polymer chains. So if this is an glucose unit with a lot of substituents, and the white one here is with lower substituents, then one can then look into how random this substitution pattern is, or how heterogeneous it is. So here we can see we have a larger part that is non-substituted than a part that is substituted. One can also talk about theoretically about blockwise substitution patterns, but here that is very rare and not used so much in the pharmaceutical, at least not for hydrophilic matrix tablets. Of course, one use blockopolymers, but not for these purposes. So we will compare these two cases here, random and heterogeneous distribution. And here we have the heterogeneous, the drug release of carbamazepine from the heterogeneous one, and here we have from one homogeneous one. And you can see that the difference in release rate is 13 hours here. That is a huge problem. If you start to develop a drug formulation using this polymer, and then you get in another batch where you get this, then the whole development is gone. So it is essential to have control over the substitution pattern. What surprised me a little bit is that still, when you buy HPMC, you buy it on degree of substitution and you buy it on viscosity, and not on the substitution pattern, even if we know that this has a huge influence on the drug release rates. So this is a parameter that I think one can develop further. I can also say that the way one determine how much, if it is heterogeneous or homogeneous, is done by using enzyme that need, for example, 3 unsubstituted units to cleave. And if they are very random, then that will not happen. So one can then compare how much glucose that one is liberated from enzymatic cleaving. And that is the similar principle where people use, for example, for hemocellulose. I should say something about also the drug release rates. So here's another polymer matrix, that is PO, the green one here. So that is a synthetic polymer. And here's just 2 model drugs. The butylparaben has low solubility, and therefore it is erosion control. So it is released in a similar rate as the polymer. So the dominating mechanism behind the release is erosion, whereas the methylparaben is high solubility, and therefore it has a higher influence of diffusion compared to the methylparaben. So therefore they get, even if everything else is exactly the same, we get 2 different release curves here. And what people very often do is that they determine the fraction of released as a function of the time and raised to an exponential factor here. And if you have flat surfaces, the M factor should be 0.5 if it is completely diffusion control, and approximately equal to 1 if it is erosion control. So very often you can see in release studies that they have determined this factor here. So a little bit conclusion here, the API release on the water solubility of the drug, but also the size. I have not talked about the size, but if you have this same solubility but different sizes, then the diffusion coefficient will be lower, and therefore the transport will be lower. But what I have been focusing a lot in my research is on the polymer and the properties of the polymer. So there can be the molecular weight, there can be the degree of substitution, the substitution pattern, and all these has an influence on the drug release rate. So if I should sum up a little bit here, I have not talked about this one, but this statement is valid for that one as well. I didn't have time to talk about it. So oral control drug release always starts with that water is transported into the formulation, and then the API is dissolved and transported out from formulation, and that is what we call released. And now, so what is the similarities now? Yeah, going back to here, I think I have at least talked about the water transport, and that convince you that even tablets contain cellulose and that the pore structure are important. But we will soon have breakout rooms, so I would like to put this knowledge we have from the pharmaceutical industry and see if we can be of any use also in the WWSC and research. So it's quite common that one can see electronic devices that want to have different areas between the acceptor and donors, and then they create them by spinodal decomposition. I have not seen so much about that in WWSC, but one can, in the breakout room, talk about, if this is interesting, how one can use formulation or processes to control the spinodal decomposition to create different structures, for example, for electronic devices. Another one is that when we do pulping, we need to have transport of water and ice into the wood to dissolve the wood components. And then we have both diffusion control and non-diffusion control and osmotic pressure-driven transport phenomena. That is also something that is very interesting and one can dig into. Another thing, if you have that interest, is the transport of the wood components out from the wood, where many undergo plasticization, dissolution and plasticization, I should say, and transport and then the release out of it. For example, lignin is one example here. And also, we know that the nature has polymer structure that are very heterogeneous. For example, the hemicellulosas and the lignin, of course, and they also will influence the mass transport events. So these are examples of things. If you want, you can discuss them. But before that, I would like to thank the PhD students I have had the pleasure to work with. I have worked with more PhD students, but these are the one that has directly an input on the release and drug release and polymer behavior. And of course, I would like to thank all the co-authors. And I have been involved in 2 Vinnova centers, CAP that was located in Lund, and SUMO that was located here at Chalmers. We have also got I have also got money from VR and KIV for Rogin here that was looking into the lignin transport. And of course, I also would like to thank you for your attention. And by that, I think it's time to create the breakout rooms, Mikael.
Yes, thanks a lot for this very pedagogic lecture. So this is first time we do this type of thing, so we will try to. What do you say, Linda? How many groups would we do this?
Maybe 5.
Okay. I see some people dropping off, but and how long should we take it? 10 minutes, 15 minutes?
15, I think.
Sorry?
15 maybe, at
Yeah.
least. Yeah. So maybe Anette, could you those suggestions for discussion points that you had, maybe you can put them in the chat.
Yep, I can. And then, but or you can screen dump them. I stop share here. I mean, this was just points. I don't. You can discuss whatever you want, of course.
Yes, of course.
I think we should have fewer groups maybe now.
Yes, I'm just gonna copy the.
So you can copy them and then have them when you go out to the groups.
Yes. So maybe we should make 4 rooms then. I will do that in just a second. Yes.
And of course, I also answer questions if you have any questions directly to me.
I think when we come back, we can have a.
Yeah,
absolutely. And you can stop.
So let's take 15 minutes and then we come back to the main room. Okay, I will assign you, or let's see. I hope this will be fine. I think you should now have been invited.
Slutar du att spela in då?